The media coverage I looked into is about the immune system and COVID-19. The message it sends is that research have shown that the antivirus responses may last for at least 6 months after first infection of COVID, in most people, and in years in some cases. Furthermore, immune system would even become more potent after 6 months to deal with mutated versions of the virus. These discoveries show the effectiveness and effective period of vaccination. The original research report was produced by viral immunologist Giulio Lorenzi and his team at the Rockefeller University in New York City. The research was produced to find out the nature and quality of the memory B cells, which is a type of cells that are called upon to produce antibodies upon reinfection. By understanding memory B cells, scientists would be able to confirm the effective period of natural immunity. The method used in the research is case sampling and comparison. The research involved the study of 100 individuals who have recovered from the COVID-19. After 40 days of their initial infection, the researchers would use a method called RT-PCR to confirm the individuals have developed antibody responses. Then, approximately 6 months after the onset of symptoms, the individuals are called upon a second study visit to test their antibody responses. The participants were adults aged 18 to 76 and were either diagnosed with COVID infection by RT-PCR or were close contacts with someone who had been diagnosed with COVID infection by RT-PCR. In between the two studies, 13 of the 100 individuals did not seroconvert, meaning that the antibodies were no longer detectable in their blood. Thus, they were excluded from the test. Participants presented for blood sample collection and were asked to recall the symptoms and severity of clinical presentation during the first 6 weeks and the convalescent 7 weeks until second study visit phase of COVID-19. Using comparison method, researchers found that all types of antibodies decreased in number after 6 months, especially in the individuals that had higher initial levels. The decrease was more drastic. Notably, participants with persistent post-acute symptoms had significantly higher antibody levels in both studies. In correspondence, neutralizing activity also decreased over 6 months. Therefore, scientists concluded that antibody and neutralizing activity decreased significantly over 6 months, but remained detectable. This may sound like bad news, but scientists also found out that the memory B cells that are crucial for reproduction of antibody had an increase over 6 months in 21 randomly selected participants. To determine whether there were changes in the antibodies produced by memory B cells after 6.2 months, scientists obtained 532 paired antibody chains from the same 6 individuals that were examined at the earlier time point. Result shows that there were no significant changes between 2 times, but 2 of the genes in memory B cells that are significant in combating with COVID have mutated after 6.2 months. In addition, scientists also found expanded clones of memory B cells, but expanded clones of memory B cells that were found in the first experiment were no longer detectable, and new clones appeared. If we only count the number difference, the number in total expanded clone actually decreased. In simple words, although the number of antibodies decreased, they become more potent. Just by knowing these genes are crucial to combat with COVID is not enough. Scientists have to confirm the potency is indeed effective. To test this, the researchers extract the antibodies from participants and used control and mutant receptor binding domain, which is the spikes on the viruses, to test whether the antibodies would bind to them. By binding to the spikes, antibodies would be able to stop virus from binding to healthy cells in our body. The result shows that out of 52 antibody clonal pairs appearing at both time points, 43, about 83%, showed overall increased binding to mutant spikes at the 6.2 month time point. The last step for antibodies to defeat virus is when after antibodies bind to their spikes, they must neutralize it. So next, researchers test the neutralization ability of the antibodies at six months time point. Result shows that the neutralization ability at 6 month point have improved. As we know, antigen would cause our body to produce antibodies against its source. If antigen stays in the body for a prolonged period of time, it will also cause antibody to evolve to deal with its source better. And residual coronavirus tend to stay in patients' nose, lungs, and intestines even long time after a patient is cured. To find out the reason why antibodies against COVID evolved in 6 months, researchers conducted another test to see if there is residual virus in the intestine of the participants. They obtained biopsies from the upper and lower intestine tract of 14 individuals with an average of 4 months after initial COVID diagnosis, and used a variety of detection technology to locate residual virus. In the sets of detection, 8 out of 14 people have been found with the residual virus in their intestines. However, scientists were unable to be certain that the reason for antibody evolution is due to the residual virus. Finally, they have concluded that memory responses in B cells are responsible for protection and from reinfection and are essential for effective vaccination. The observation that memory B cell responses do not decay after 6.2 months, but instead continue to evolve, is strongly suggestive that individuals who are infected with COVID could mount a rapid and effective response to the virus upon re-exposure. The media coverage on this research report is generally delivers the message, but has a suspicious over-optimistic tone. For example, the coverage frequently suggests how strong our immune system will be 6 months after exposed to coronavirus, using words like people appear to have built an arsenal of antibodies that are not only more potent than the ones developed early on. This may be due to the fact that an optimistic tone fits public taste, especially when everyone has suffered so long from this pandemic. But the strength of immune system varies in different individuals. And an important premise for immune system to evolve is that people could be effectively treated and survive after initial infection. The study is conducted to test the effective period and involvement of natural immune system after individuals being infected with COVID, and only hints on the effectiveness of vaccination. But the media coverage almost totally shifts the perspective to vaccination. To determine whether a vaccination would have the same effects as natural immunity, further studies must be conducted. And the effectiveness of memory B cells is only one crucial factor to confirm the effectiveness of vaccination. Furthermore, the media coverage says that immunity from a vaccine is expected to last as long or longer than natural immunity, which is totally out of the scope of this research report. In addition, the media coverage contains some data error, like 7 of 14 recovered COVID-19 patients had evidence of coronaviruses in their intestinal tissue. But in the study, it is in fact 8 out of 14.